Welcome to the Deep Dive, the shortcut you needed into the most exciting complex topics in science. Today, we aren't just managing health, we're exploring how to rebuild it. We're diving into regenerative medicine, humanity's quest, really, to achieve true healing, replacement, and functional restoration. We're trying to move beyond simple maintenance and set our sights much higher on actual biological repair. Okay, let's unpack this. We have a stack of fascinating sources today that give us a vital map into this field. If you are preparing for a meeting, or maybe you simply want to understand how a damaged organ could potentially be replaced by a healthy, lab-grown counterpart, well, this deep dive is your guide.
Yeah, and regenerative medicine at its core, it's this burgeoning field that leverages cellular power. It's quite amazing, really.
Right.
We're talking about the ability to identify, isolate, expand, and then precisely transplant stem cells and their differentiated progeny to achieve restoration. The goal is transformative, to repopulate damaged tissues or maybe even construct entire functional replacement organs.
And our mission today is, well, it's ambitious. We want to move well beyond the exciting headlines, We want to truly understand the fundamental biology, the cell cycle, the whole concept of cell identity, and the stem cell's unique power that underpins this entire endeavor. Because the precision required to fix a damaged heart, it starts right down at the level of controlling the machinery of a single cell.
Absolutely. And this is intensely relevant right now because regenerative medicine offers therapeutic opportunities for tissues that currently, well, they fail us most spectacularly, precisely because they have such low intrinsic regenerative capacity. We're talking about the tissues that just cannot repair themselves after catastrophic damage. Think the heart after a myocardial infarction, a heart attack, or the brain after a stroke. This science aims to restore the function lost in those events. It represents a total paradigm shift from long-term supportive care to genuine biological restoration.
Okay, so before we can even talk about regenerating an entire human organ, like you said, we have to grasp the absolute basics of self-maintenance. How does a tissue keep itself healthy day to day, that constant ongoing battle?
Homeostasis, right?
That's the critical starting point. Cell proliferation is really the engine that drives this maintenance. It's fundamental. It's required for development, for the daily steady-state maintenance of tissue homeostasis, and crucially for replacing cells that naturally die off or get damaged. If the rate of production doesn't quite match the rate of loss, the tissue fails. Simple as that.
And that cellular engine, it runs on the cell cycle. We all learn the phases in school, G1, S, G2, and M, but for what we're talking about today, we need to know that this is a system built not really for speed, but for incredibly regulated precision. It is a meticulously regulated sequence. G1, that's the initial phase of growth. S is where the critical task of accurate DNA synthesis occurs, replicating the entire genome, flawlessly, hopefully. G2 is the preparation for division, and M is the actual mitotic phase where the cell divides. But perhaps the most important phase for understanding regenerative medicine, interestingly, is G0, the quiescent state.
G0, why G0 specifically?
Well, G0s are like the default waiting room for cells. Many stable cells, think hepatocytes in the liver or fibroblasts in connective tissue, they're normally just resting in G0. They're functionally active, doing their jobs, but not actively proliferating. Yet, crucially, they retain the capacity to re-enter the cycle and divide rapidly if they receive a specific signal, like an injury. They're ready to be called up from the bench, so to speak.
But then we have the problem children, right? The permanent cells.
Exactly. Permanent cells, like most mature neurons and cardiac myocytes, heart muscle cells, they've effectively lost the capacity to proliferate altogether. They've been forced into a deep, seemingly irreversible G0 state. And this is precisely why damage to the heart muscle or brain tissue is so permanent and devastating. Regenerative medicine exists primarily because we need a way to somehow override this permanent off switch in those cells.
Okay, it sounds like a relay race, but with extremely complex rules and quality control checkpoints all along the way. What governs the pace of this race and makes sure cells don't replicate flawed DNA, especially when we're trying to force proliferation in regenerative therapies?
That control system, it involves the cyclins, the cyclin-dependent kinases, CDKs, and their corresponding inhibitors, the CDKIs. Think of the entire system like a car, maybe. The CDKs are the engine. They're enzymes that phosphorylate substrates. They push the process forward. But the CDKs are useless on their own. They have to bind to a cyclin.
So the cyclins are the key and maybe the accelerator pedal.
They're the timing mechanism, precisely. Cyclins are proteins whose production and degradation are strictly cyclic. They rise and fall predictably across the phases. You have cyclins D, E, A, and B appearing sequentially. When a specific cyclin transiently increases, it binds to and activates a specific CDK, pushing the cell to the next checkpoint. And it's the duration and amplitude of this cyclin activity that dictates critical cell fate decisions. Not just whether the cell divides, but also whether it remains a stem cell or commits to differentiation. It's very finely tuned.
And just as important as the accelerator, we obviously need the brakes. And those quality control centers you mentioned.
That is the role of the cellular checkpoints and the CDK inhibitors. We have the G1S checkpoint, which is arguably the most crucial. It monitors DNA integrity before the cell commits massive resources to replication. We also have the G2M checkpoint, ensuring accurate genetic replication before the cell physically divides.
And if those checkpoints detect a problem, a mutation or something, or if the cell is one of those permanent tissue types, how is the stop signal transmitted?
That's where the brakes, the CDKIs, step in. They modulate, or sometimes outright inhibit, the CDK cyclin complex activity. We have two major families. One family, including P21, P27, and P57, broadly inhibits multiple CDKs. They're like general brakes. The second family, exemplified by proteins like P15 and T16, selectively targets the cyclin D CDK46 complex, and that complex is critical for moving out of G0 and back into G1.
Ah, okay. And here is the connection back to regeneration, right? If a cell detects serious damage, the CDKIs trigger either repair or they enforce a permanent shutdown.
Exactly. If repair fails, the cell is often forced into programmed cell death, which we call apoptosis. Or, alternatively, it enters senescence. Senescence is a permanent, non-replicative state, often enforced by proteins like P16. Now, this is critical for preventing tumor formation, which is good, but it's also a major roadblock to regenerating aged or damaged permanent tissues like the heart or brain. So, the job of regenerative medicine, when treating the heart, for instance, is to figure out how to safely and transiently disable those brakes, like P16 or maybe P53, to coax those quiescent cells back into G1. But without inadvertently triggering cancer. That's the tightrope walk.
That is a remarkable insight. We're literally talking about using small molecules, drugs, to artificially release brakes that evolution put in place for our protection. This whole need for controlled proliferation brings us directly to the star of the show, the stem cell.
Yes, stem cells. They possess that crucial dual property we desperately need for functional regeneration. First, the ability to self-renew indefinitely, making more of themselves. And second, the ability to give rise to differentiated cells and tissues, the specialized cells we need. In healthy tissue, this is usually a delicate homeostatic equilibrium. Stem cell replication and differentiation are perfectly balanced against the necessary apoptosis, or death, of mature, spent cells.
And the type of division the stem cell undergoes actually dictates how this balance is maintained.
Yes, precisely. For routine daily maintenance, the cell primarily utilizes asymmetric division. This is the most common way they split in adult tissues. What happens is one daughter cell enters a differentiation pathway. It commits to becoming a specialized cell needed for tissue replacement, while the other daughter remains undifferentiated, conserving its self-renewal capacity. So you get one functional replacement cell, and crucially, you keep the reserve stem cell pool intact.
One for function, one for the future. Got it.
Exactly. The other mode is symmetric division. Here, both daughter cells retain that self-renewal capacity. Now, this is much rarer in adult tissues. It happens primarily when the body needs to aggressively expand the stem cell pool itself. For example, during the intense growth of early embryogenesis, or perhaps when recovering from extreme depletion, like bone marrow repopulation after ablative chemotherapy destroys the existing pool. The difference between these two modes, asymmetric and symmetric, is really the difference between maintenance and expansion. And controlling which mode happens when feeder is absolutely central to successful cell therapy.
Okay, now that we understand the power source that controlled proliferation, let's look at the actual building blocks themselves. Our sources clearly lay out the two fundamental varieties of stem cells, defined mainly by their origin and their sheer power, or potency. Here's where it gets really interesting, I think.
The distinction is absolutely crucial because it dictates the risk, the ethical constraints involved, and the therapeutic scope of the cells we might use. We can think of it as moving along a spectrum, starting with the most powerful, the totipotent cells, and going down to the more restricted, multipotent cells.
Right. Let's start at the top then with embryonic stem, ES cells. What are they?
ES cells represent the apex of potential. They are harvested from the inner cell mass of the blastocyst. That's a structure formed just a few days after fertilization, very early in development. Their power is, well, it's almost limitless. They're considered totipotent. That means they possess virtual immortality in culture, they can keep dividing, and they can give rise to every single cell type in the body. That includes cells from all three primary germ layers, the ectoderm, mesoderm, and endoderm.
Wow. That's like a universal cellular toolkit. So what's the practical application of this immense potential in the lab?
It means that, provided we find the perfect cocktail of growth factors and specific culture conditions, which is a big if sometimes, scientists can induce these ES cells to form pretty much any highly specialized cell type we need. Fully functional neurons, insulin-producing beta cells for diabetes, or even beating cardiomyocytes to repair the heart. They are the ultimate biological raw material, capable of forming everything. But, and this is a big but, their source introduces significant ethical debates and clinical logistical challenges, particularly immune rejection.
Which naturally leads us to the cells doing the daily grind in our bodies. The tissue stem cells, often called adult stem cells.
Correct. These adult stem cells are found nestled within the mature tissues they maintain throughout life. Unlike ES cells, their capacity is much more restricted. They're typically multipotent, not totipotent. This means they can usually only generate the specific cell types found within their residing tissue. So, a blood stem cell makes blood cells, an epithelial stem cell makes skin or gut lining cells. They're more like a specialized repair crew, not those universal contractors like ES cells.
And you mentioned earlier they don't just float around unprotected. They need a highly specialized environment, a niche.
Absolutely. They are protected within these specialized microenvironments called stem cell niches. You can think of the niche as the stem cell's highly regulated private office, or maybe its support system. It's a precise physical location defined by its structural components, things like the extracellular matrix, but also by local soluble factors, growth factors, cytokines, and crucially, by the surrounding support cells, like vascular cells or fibroblasts.
So the niche isn't just a location, it's an active signaling system.
Precisely. The niche provides the constant, nuanced signals that regulate the stem cell's behavior, its balance. It dictates whether the cell remains in quiescence, that's the do not disturb G0 state we talked about, whether it should aggressively expand its numbers, or whether it should begin the differentiation process to replace damaged cells nearby. And importantly, if the niche itself is damaged or disrupted, say by massive inflammation or fibrosis following an injury, then the stem cell function often fails, and the tissue can no longer regenerate efficiently. The niche is critical.
Okay, let's make this more concrete. Can you give us the most clinically relevant examples of these adult stem cells and their niches, like HSCs and MSCs?
Sure. The hematopoietic stem cells, HSCs, are probably the most famous and definitely the most utilized clinically today. They live primarily in the bone marrow niche, although they can circulate too. And their job is to continuously replenish all the cellular elements of our blood red cells, white cells, platelets, everything. Clinical application is standard practice now. We use HSC transplants, bone marrow transplants to repopulate marrow that's been depleted after aggressive chemotherapy, often necessary for treating leukemia, for example. Or they can be used to correct severe genetic blood disorders like sickle cell disease or thalassemia.
Okay, HSCs for blood. And the mesenchymal stem cells, MSCs, they seem to be the favorites for structural repair and this whole bioengineering angle.
They really are the structural workhorses. MSCs are found in bone marrow alongside HSCs, but also in adipose tissue, fat, and other connective tissues. They are multipotent, meaning they can differentiate into various stromal cell types, chondrocytes for cartilage, osteocytes for bone, adipocytes for fat, and even myocytes for muscle. Their natural role in building and repairing structural tissue makes them incredibly important for orthopedics and regenerative surgery.
And crucially, our sources emphasize that MSCs hold a major advantage over ES cells when it comes to the immune system. This seems to address one of the biggest challenges in the whole field.
Yes, it's a potential game changer, though maybe not quite as simple as initially hoped. MSCs can be massively expanded in culture, which is useful for generating large numbers. But their real potential therapeutic value lies in their ability to generate a locally immunosuppressive microenvironment. They achieve this by secreting specific soluble factors, molecules like prostaglandin E2, PGE2, and indolein E2-folate3 dioxygenase, IDO. These molecules actively suppress the local activity of T cells and other immune effector cells that would normally attack foreign tissue.
So they actively tell the local immune system to sort of stand down, calm down.
Effectively, yes. They modulate the immune response locally. This grants MSCs what we call relative immunological privilege. It minimizes, though doesn't always eliminate, the need for complex HLA matching between donor and recipient, and allows them to be explored, in some cases, as an off-the-shelf type therapy. This property makes MSCs the primary candidates for manufacturing large-scale structural cellular scaffoldings, allowing us to potentially build replacement tissues like bone grafts or cartilage patches with a minimized risk of immediate rejection.
Okay, the theoretical potential is clear. We have these universal building blocks like ES cells and the specialized structural cells like MSCs with some immune advantages. But when we try to step into the clinic, what stops us from simply transplanting these lab-grown cells right now, widely? We mentioned two big persistent hurdles.
Indeed, these two difficulties really represent the gap, the challenge in transitioning from basic science discovery to scalable patient therapy that actually works reliably.
Let's talk more about the first one, the physical hurdle, the integration challenge. What does that really entail?
This is a really complex bioengineering problem at its heart. We're not just trying to inject a functional cell type into damaged tissue. We need that cell to first survive the often hostile environment of the injury site, the inflammation, lack of oxygen, and then, most critically, we need it to functionally integrate into the existing host structure. For example, if we inject stem cell-derived cardiomyocytes into the scar tissue of a heart after a heart attack, they need to live, yes, but they also need to align structurally with the existing muscle fibers. And absolutely, essentially, they need to electrically couple with the billions of existing host cells so they can beat in perfect unison. Simply surviving isn't enough. They have to join the choir and sing the same song at the same time. That's incredibly hard.
Right. And the second hurdle is the one we touched on with ES cells, particularly
Right.
the immune system's relentless defense mechanism, immunologic rejection.
This is the body's ultimate ID check system. All nucleated cells, including stem cells and their derivatives, express proteins on their surface called histocompatibility molecules. In humans, these are primarily the human leukocyte antigen HLA system molecules. If you transplant cells derived from a non-patient source, what we call allogeneic cells, the host immune system recognizes these different HLA molecules as foreign, as non-self, and it mounts a vigorous, often destructive rejection response. This forces us, typically, to use powerful, lifelong immunosuppressive drugs which carry their own severe risks, like infection and cancer.
Okay, so we have the universal building blocks, ES cells, but they get universally rejected. We have the specialized structural cells, MSCs, which are somewhat privileged immunologically, but they don't have the immense power needed to, say, rebuild an entire complex organ like the brain. This deadlock, this challenge, it really forced scientists to search for a new type of building block, a patient-specific one, which led to this incredible creation of induced pluripotent stem iPS cells.
Exactly. The development of iPS cells by Shinya Yamanaka's group, which won the Nobel Prize, was such a breakthrough precisely because it offered a way to potentially solve the immunological rejection problem while retaining the necessary pluripotent power of ES cells. The solution was reprogramming this idea of convincing a fully differentiated mature cell, like a skin cell, to fundamentally reverse its identity and return to an embryonic-like pluripotent state.
It really does sound like biological time travel. How is this dramatic reversal actually achieved? How do you convince a skin cell it's young again?
It demonstrates a remarkable cellular malleability we didn't fully appreciate before. Scientists identified a specific handful of transcription factor genes, often just 4, and they're referred to as the Yamanaka factors, OCT4, SOX2, KLF4C in my case. They introduce these genes into easily accessible, fully differentiated somatic cells, typically fibroblasts harvested from a simple patient skin biopsy. When these genes are expressed, even transiently, they essentially rewrite the cell's genetic operating system. They manage to override those CDKIs we talked about. They reset the epigenetic markers that define cell identity, and they remarkably reprogram the somatic cells to achieve the stemness, the pluripotency of ES cells.
And because these iPS cells are derived directly from the patient's own tissue, what is the revolutionary clinical consequence of that?
The huge consequence is that any new tissue generated, the differentiated progeny of those iPS cells, can theoretically be transplanted back into the patient without fear of major immunologic graft rejection. They carry the patient's exact HLA markers. The immune system sees them as self. No need for those dangerous immunosuppressive drugs.
Okay, this sounds like the perfect solution. ES cell potential combined with perfect immune compatibility. But wait a second, I have a fundamental concern here, and I'm sure you, the listener, might too. If a patient has an underlying genetic disease, say a form of inherited muscular dystrophy or maybe type 1 diabetes with a genetic component, and we take a skin cell from that patient to create iPS cells, aren't we just creating brand new, compatible, but still inherently diseased cells? Doesn't that kind of negate the whole point for genetic disorders?
That is an excellent and critically important question that really pushes the discussion forward into the next level of application. And the answer is twofold. First, even generating diseased tissue in this way allows researchers to study the disease mechanism in a dish using patient-specific cells. That's incredibly valuable for understanding the disease and testing drugs disease in a dish models. Second, and more importantly for actual therapy, iPS cell technology is now being combined synergistically with modern gene editing techniques.
Ah, okay. So we fix the genetic error in the iPS cells before we even think about transplanting them back.
Precisely. That's the workflow. You harvest the somatic cell from the patient. You create the iPS cell line, which carries the disease mutation. You then use incredibly precise tools like CRISPR-Cas9 to go into the iPS cell's genome and correct the underlying genetic mutation, fixing the mistake. Once corrected, you then differentiate these now healthy, genetically repaired, and patient-compatible iPS cells into the required tissue type, say, healthy muscle cells or healthy neurons, and only then do you transplant them back into the patient. This integrated approach reprogramming plus genetic correction represents probably the most powerful therapeutic promise in regenerative medicine today, especially for inherited diseases.
That brings us right back to the source materials example. Taking a diabetic patient's skin fibroblast, maybe correcting any genetic predisposition to diabetes in the iPS cells, differentiating them into healthy, insulin-secreting beta cells, and transplanting them back. No rejection, and potentially no more disease. That truly is the vision of personalized restorative medicine. Okay, that brings us right up to the current state of play, more or less. The blueprints are drawn, the biological mechanisms are largely understood, but the practical progress, the engineering, is what determines how quickly this hits widespread medical practice.
Absolutely. And the scientific community has really doubled down on refining the technologies that minimize rejection risk and maximize potential. That means most effort right now is focused on advancing MSCs for their immune properties and structural uses, and iPS cells for their patient-specific pluripotency.
Right. Let's analyze the continued development of iPS cells first, then. While the potential is obviously huge, the concept of turning a mature cell back into a state of near embryonic potential, it sounds inherently risky, doesn't it?
Yeah.
What is the critical safety concern the field is grappling with right now?
The critical safety concern, the big one, is tumor formation, specifically the risk of forming teratomas. If even a tiny fraction of the transplanted iPS cell-derived population fails to fully differentiate, if some cells retain their pluripotency, that ability to become anything, they can differentiate uncontrollably at the site of injection. This can result in a teratoma, which is a benign but disorganized mass containing various tissues like bone, hair, muscle, neuronal tissue. It's highly undesirable, obviously. So current advances are focused intensely on refining the generation and differentiation protocols to achieve ideally 100% efficient and specific differentiation into the target cell type only. No pluripotent stowaways.
So the main focus is on a perfect conversion rate, making absolutely sure no undifferentiated stem cells slip through the net into the patient. How are they also improving the efficiency and safety of the reprogramming process itself, getting the skin cell to become an iPS cell in the first place?
Yeah, that's also critical. Early methods often relied on using retroviruses to deliver those Yamanaka factor genes.
Yeah.
The problem is, retroviruses can permanently integrate the reprogramming genes into the host cell's genome. That integration can happen randomly and potentially disrupt tumor suppressor genes or activate oncogenes, significantly raising the long-term cancer risk. Not ideal. So today, the field is moving rapidly towards much safer non-integrating methods. These include techniques using messenger RNA, mRNA, some RNA, like some COVID vaccines, or modified non-integrating viral vectors like Sendai virus. These deliver the reprogramming factors transiently, they do their job, and then they disappear without permanently altering the host DNA.
Yeah.
This refining of delivery methods is absolutely key to scaling iPS technology safely for clinical use.
Okay, let's shift back to the structural engineers, the MSCs. We talked about their relative immune privilege and their potential for creating structural scaffolding. How is bioengineering actually turning this theoretical advantage into physical, implantable tissue constructs?
This is where tissue engineering really merges beautifully with cell biology. Researchers are actively leveraging the osteogenic bone-forming and chondrogenic cartilage-forming properties of MSCs, and they're combining them with advanced biomaterials, things like hydrogels, synthetic polymers, or even decellularized tissues from donors which act as natural scaffolds. These materials are engineered to act as three-dimensional scaffolds. essentially precise molds designed to mimic the complexity and structure of the native extracellular matrix in bone or cartilage.
So the biomaterial scaffold dictates the final shape, provides the structure, and the MSCs seated onto it do the biological filling and building.
Exactly. The scaffold guides the MSCs to proliferate, differentiate correctly, and organize themselves into complex functional shapes. Maybe a custom piece of replacement cartilage, perfectly shaped for an injured knee joint, or perhaps a large segment of replacement bone for a fracture that won't heal. And because the MSCs themselves are locally immunosuppressive, the whole bioengineered construct has a much better chance of successful integration and eventual remodeling by the host's own cells once implanted into the patient.
We're really moving from just injecting suspensions of undefined cells towards manufacturing preformed, potentially load-bearing replacement tissues in the lab.
And finally, let's revisit those really ambitious targets, the heart and the brain. We established that the integration challenge is massive for these tissues full of permanent, non-dividing cells. Are clinical trials showing any real progress in getting these complex replacement cells to, as you put it, sing the same song as the host tissue? Progress is definitely being made, but it honestly remains the steepest mountain to climb in the field. Clinical trials utilizing various types of stem cells, including those derived from iPS cells, targeting myocardial damage after a heart attack or neuronal damage after stroke, are certainly ongoing in many centers. The key challenge remains not just cell survival, but achieving that robust functional integration we discussed earlier. For the heart, for example, researchers are now focusing heavily on methods to ensure proper electrical coupling between the transplanted cells and the native heart muscle. This sometimes involves using engineered tissue patches that incorporate conductive biomaterials, trying to bridge the electrical gap and ensure that the transplanted cells fire perfectly in sync with the native myocardium. It's incredibly complex.
It really sounds like the solution isn't just a biological one anymore, but a convergence of biology and engineering. The sources definitely imply that this convergence is the future.
That is absolutely the core message emerging now. The construction of functional replacement tissues, even complex ones, is becoming a truly foreseeable reality, not just science fiction. And it's happening because we are achieving an unprecedented convergence. We're mastering the fundamental cell cycle controls. We're utilizing sophisticated genetic tools like iPS reprogramming and CRISPR gene editing. And we're employing advanced delivery systems and structural methods developed through bioengineering. We are learning step by step how to manually adjust the dials of self-renewal and differentiation with unparalleled precision.
So wrapping this all up, what does this all mean for you, the learner listening to this deep dive? Where does this leave us?
I think it means regenerative medicine is not just a small tweak to existing medical practice. It really represents a fundamental paradigm shift. We are genuinely moving away from merely treating symptoms or just managing biological decline towards proactively replacing lost tissue and restoring lost function. The key knowledge you need to take away, really, is the importance of controlling that unique stem cell's dual power. The power to self-renew indefinitely, making copies, and the power to differentiate precisely on command into the needed cell type.
And let's just quickly solidify the key players again in this new biological arsenal we've discussed.
Okay, we have the embryonic stem, ES cells, which possess that universal totipotent potential but face significant ethical and rejection challenges. We have the adult stem cells, like the HSCs in bone marrow and the MSCs in connective tissue, which handle tissue-specific maintenance, protected and tightly controlled by their specialized niche. And critically, we now have the induced pluripotent stem iPS cells. These provide the patient-specific solution, utilizing genetic reprogramming to turn a simple somatic cell, like a skin cell, into an immune-compatible, powerful building block for personalized repair.
And that really leads us to the final provocative thought for you to chew on after this deep dive. The sheer ability to reprogram a fully differentiated cell, a skin cell, a fibroblast, cells we thought were fixed in their identity back to its embryonic-like pluripotent state, as we do routinely now with iPS cells. It fundamentally suggests that cellular identity is far more fluid, far more malleable than scientists ever dreamed possible just a few decades ago. If we can chemically and genetically convince a mature skin cell that it's actually an electrically coupled beating heart cell or a dopamine-producing neuron, what limits truly remain on treating virtually any genetic or acquired disease where cells are lost or damaged? The science of the impossible is rapidly becoming the science of the inevitable.
The major challenges now are moving towards industrial scale production, ensuring absolute safety and efficacy in large trials, and figuring out the clinical logistics and cost for widespread distribution. But the fundamental biological pathway to functional healing, It feels like it's finally laid bare.
We definitely encourage you to continue exploring the fascinating molecular pathways that govern pluripotency and also the exciting, sometimes complicated intersection of gene editing and stem cell therapies. It's where so much is happening. Thank you for joining us on this deep dive into the future of health.